Now, one thing about some of the things that I'll be talking about today that is sometimes not very appreciated is just an enormous amount of expertise and that needs to go into the work that I'll be describing. So I'm a neuroscientist. I'm a cognitive neuroscientist. I study brain development in Portland, Oregon at Oregon Health and Science University. And I'll be talking about my work, but it's not done in isolation. We have lots of folks who are clinical psychologists who help with diagnosing and assisting with trying to understand the phenotypes of individuals, children. We have lots of folks from even social workers that are down here at the end. We have lots of students and postdocs and just an enormous amount of people that are required to be able to do the types of work that we do and to really to advance the kind of research that I'll be describing shortly. So I always like to acknowledge and just point that out before I start this presentation or this workshop. All right, so there are four main goals that I want to talk to everybody about today. One is about this new methodology we've been using that I think is going to be extremely powerful as the years move on, called functional connectivity, MRI. The second goal is I want to discuss what we've been dubbing the heterogeneity problem in developing populations and how that's going to be really important for education and clinical work moving forward. I describe another mathematical discipline that we're using to try to understand how the brain develops, called graph theory, and then I'll give some brief examples of how all this stuff kind of combines together to give us a better understanding about children and brain development. So I'm going to start here with this functional connectivity MRI business. All right, so most people have heard of, at this point, in the popular press or in other media, what traditional fMRI is. In this case, what we typically do is we have any subject go into a brain scanner that looks much like this here, and we have them do some task that's interleaved with some type of control task. So in this case, what I'm showing here is someone going into the MRI scanner and they open their eyes and then they close their eyes. And they open their eyes and they close their eyes. And they open their eyes and then they close their eyes over and over and over and over and over again. We do some selective averaging of the signal that comes out of the MRI scanner. And then what we're able to see is parts of the brain that are active when your eyes are open versus when your eyes are closed. Now, lots of folks always ask me in clinic, well, why is it that you have to have the participants when they're scanner open their eyes and close their eyes so many different times? Why can't you just have them do it once and be done? Why do we have to open and close and open and close and open and close and open and close over and over and over again? And the reason is because the signal that comes out of the MRI scanner is very noisy, meaning it's got lots of what we thought for a long time, a lot of misinformation in there. And so the example I just gave a talk similar to this to Congress, and the example I gave them was that kind of that made sense was polling data, right? So when you poll a lot of different time points like this, sometimes the polling can be very messy over and over. And that means it's because there's lots of noise in the signal. It depends on any given number, depends on who you polled and what time of day it was and all the different stuff. So we do either we average over lots of different polls or we average over lots of period of time to help smooth out the data to make it make more sense. And we do the exact same thing when we do fMRI. Now, in 1995, this guy who we know now, his name is Berger, decided to look a little bit more at what we always thought was noise, this kind of misinformation that was in the scanner that we had to average out. And what he did was he had people go in the scanner and he had them do this finger tapping task. Instead of this time, they would tap their fingers and then rest, and they would tap their fingers and then rest, and tap their fingers and rest, over and over and over and over again. And they do the selective averaging, and then lo and behold, the parts of the brain that were important for moving your fingers became more active when you were tapping them versus when you were not doing anything at all. Okay? But then he took the experiment one step further, and what he did was he had the same people come back in the scanner, and this time what he did is he just had him sit there and not do anything at all. Okay? He would just have him sit in the scanner, don't tap your fingers, don't do anything at all. And he took one of these brain regions and just calculated all that noise that kind of just happens over time. And he correlated that noise with the rest of the brain. And lo and behold, what he found was that even though the participants aren't tapping their fingers, the same parts of the brain that were active when you were tapping your finger, even when you're not doing anything at all, are sitting there spontaneously oscillating with each other when you're at rest. And so what that told us is that this stuff that we would even kind of remove out in order to see the activity isn't really misinformation or noise at all, but it's actually functionally relevant brain signals that are giving us information about how the brain is organized intrinsically. Okay? And this is in essence what we call resting state functional connectivity. And this is one of the methodologies that's being used increasingly over the last several years to really help us understand how the brain works. Now, folks always ask me, well, why is it, who cares about the brain when we're resting, right? When we have someone doing a task or trying to pay attention or trying to memorize something or doing something, they're always doing something, right? Well, probably one of the biggest arguments of why this is really important comes from the work in metabolism, or what we like to call the energy budget. So, everybody just had lunch, right? And even though the brain is only about 2% of the total body size or body weight, it consumes over 20% of the energy. So of that food you just ate just now, 20% of all the energy is going to go straight to the brain. Anything that comes out of it is going to go straight to the brain. And that's really important. It means this brain is really highly revved up, right? Now, when we do these fMRI experiments, the stuff that you see in the New York Times or in your local papers, but some new fancy experiment, that is really only a really tiny amount. So, like if when you're actually doing a task, like let's say writing a paper or grading something, the actual movements, the actual act of doing that task is reading a book, is actually very little. It consumes very little of the energy that's going on all the time. So here's just an example of this. So this is your brain activity when you're just sitting there fixating, not doing anything at all, kind of like everybody's listening to me right now. This is what it looks like when you're actually engaged in something like reading a book or viewing words. Okay? Now what you can see on this picture, or at least I can, from at least my perspective, is that you can't really tell much of a difference at all, right? They look almost identical, right? But the only reason why we get those fancy pictures in the papers or the magazines, right, with regard to fMRI experiments is because we can look at a difference between these two, and we can see how they differ, even though it's very small. Okay? So the amount of energy consumption of the parts of the brain that are important for reading relative to when you're doing nothing at all it's less than 5% of that total 20% that you just put to your brain after you're eating lunch. Okay, so 95% of all the energy that your brain uses from your lunch today is going towards the brain when you're at rest, essentially not doing anything at all. Okay, that's very important. Now, the brain does lots of things besides just making signals or doing different types of neuronal signaling. The neurons have to be repaired. We have to kind of move around different types of proteins in case you got an infection or something like that. And so when this type of work was first discovered, it was questioned whether it's really important for the actual brain work, right? And I'm not going to really go into depth about this figure, but we now have technologies with MRI where we can actually look at the chemical signaling that occurs in the brain with some of these MRI technologies, and shown that this energy budget is actually going towards neuronal signaling. Now, the other thing that folks actually ask me for, well, if it takes up all this energy, right? Well, then what is this stuff? What's it doing? What's it important for, right? And some people, there's lots of ideas, and this is stuff that we're still working out, but some people think it's really important for maintaining specific types of neural networks or facilitating communications between one part of the brain to the other part of the brain. Or things like building internal representation, so like the learning. When you learn something new, you build a representation of what it means to change a tire, for example, right? Or binding or linking things that you see in your environment to what you expect in your brain. Now, there's a couple of examples that I think that make this really salient to folks, particularly who are new or trying to learn about, how the brain works, right? So it may come to surprise to most of you that from a nearly unlimited amount of information that comes from your visual world, like the stuff that you're seeing outside of you right now, only about 10 to 10 bits per second of information actually hits the retina, your eye, right here. Okay? That's about the same resolution that you have at home on your Blu-ray DVD. That information then travels to another part of the brain that we call the thalamus, and by the time it gets back there, it's degraded down to about 6 times 10 to the 6 per second. That's about the same amount of information that a standard definition TV, the old school, does anybody even have those anymore? I don't know. Standard definition TV produces, right? By the time that information gets way back here, this is your visual cortex. This is the first part in your brain that actually sees this information. The information from your world has been degraded down to 10 to the 4th bits per second. And that's about the same amount of information that you see on the video phone. And when I was, preparing this talk, I went and looked up on, video phone resolution on Wikipedia, and this is what they say. That's the minimum necessary for a consumer acceptable talking head picture using various video compression schemes. Okay? It's something you see from the reporters that are overseas at war in Iraq or Afghanistan, right? And by the time that information gets to kind of the stuff that you're actually aware of, like you're seeing me right now, you're aware that I'm actually present, that's about 100 bits per second. That's about the amount of information that passes via your cell phone or some voice over IP like Skype or something, right? So the question is, how in the world does the brain make sense of such impoverished information to provide us with a visual experience, like right now, that some might even say is better than Blu-ray DVD? And the answer is that it's the stuff that is percolating in the background. It's all of this information, it's all of this spontaneous activity that's going along in the background. Essentially what happens is that the brain uses a bunch of energy, I'm not going to go through the details of this, uses a bunch of energy and has a lot to maintain expectations of what you should see. And then you have these very degraded signals that come in that trigger those expectations to give you a visual world that seems perfect, even though it's not there. If any of you guys have ever seen a magician do some really crazy magic, this is the reason why it works. It's because your brain has a lot of expectations that are sitting there oscillating in there all the time, and you're unable to, and if your magician can trick your brain into thinking it's seeing something, which will trigger that internal stuff, and so you'll be blind to what's really happening beside it. This is the only reason that magicians even have jobs, is because they're able to manipulate this level of activity in the brain. Okay? Now, there are some other examples of this that I sometimes like to give, and it's funny. I was giving this. I was talking to some of the teachers here on my way over on the bus, and I had this kind of really degraded, so to speak, talk to make sure that I could really focus and everybody can understand all this stuff. And as I was talking to them, I realized how sophisticated you folks really are. And so I decided I'm giving a talk just like this to the National Institute of Health and the White House in 1 week. And I decided to add all this stuff information because they were asking questions about this exact scientific thing. So I'm very impressed. So this is the exact same scientific talk that I'm going to be giving to some folks up on the hill. Now this is another example of how this spontaneous brain activity is used in things like memory, things that you guys undoubtedly examine or evaluate in kids in your classroom. So this is I'm sure folks have heard of mice going through mouse experiments, like going through a maze, right? You put a mouse through a maze, they try to see how often or if they can remember how to get through a specific maze, right? Well, one of the things that we can do with neuroscience is we can actually record neurons in part of the brain called the hippocampus that's really important for monitoring locations, okay? And this is just an example of a mouse going through a maze and recording cells in the hippocampus which kind of monitor the position the mouse is at any given time. So this is the actual position of the mouse. And this right here, we have is recordings in different cells showing that are recording this exact same position. Now what's interesting is not necessarily when the mouse goes through the maze, but actually what happens after, when they're just sitting there not doing anything at all, okay? And what happens is that these little lines here are blown up over here. And what happens is these cells, after you've gone through this maze, what happens is these cells, when you're sitting there at rest not doing anything at all, they replay that experience, except for they replay it much faster and they replay it in reverse. And so what's happening here is these things, you can see this place going this way. These little lines here are doing the exact same thing, doing it in reverse. All this stuff is happening when you're not doing anything at all. So the information that's acquired during this resting period is really, really, really important for brain function. And so I always like to have this quote. Folks way back in 1990, William James, the very, very famous psychologist, who pointed out, way long ago, we're just recognizing it now, is that what's part of what we perceive comes from the senses or the objects that are in front of us. Another part, and it may be the larger part, always comes out of our own head, okay? So this is the spontaneous brain activity I'm talking about. The other person who recognized this a long, long time ago was this man named Berger. He's the guy who invented what's called EEG. Some of you guys have probably heard of that. And he pointed out way back when, is that of course, it's possible to demonstrate intellectual work, meaning activity, on the EEG, but one should not entertain too high hopes in this regard because mental work, as explained elsewhere, adds only a very small increment to the stuff that's going on all the time.